Hello all of you. In this class we will discuss about different tests for the quantitative analysis of different amino acids. After that we will talk about the peptide and peptide bonds, and the last we will discuss about different structures of protein. So let's first start with different tests for the detection of amino acid. So in this slide, I would like to talk about different tests for the detection of amino acid in protein solution. So in this table, there are 10 tests for the amino, quantification of different amino acids. And among these two tests are important for your course. First is ninhydrin test and second is xanthoproteic test. So about ninhydrin test, I have already taught you that when amino acids react with ninhydrin reagent, they give purple color in reaction mixture. Exception is proline and hydroxyl proline, which give yellow color when they react with ninhydrin reagent. Now let's talk about another important test for the detection of amino acid, that is xanthoproteic test. And xanthoproteic test is used to detect amino acid containing aromatic side chains. So what are those amino acids? So those amino acids are tyrosine, tryptophan and phenylalanine. So in this test, when all these aromatic amino acids reacts with concentrated uh, nitric acid in presence of heat, they give yellow colored nitro derivatives. And here you will have to remember one thing, that phenylalanine gives negative or weakly positive reaction, because for this amino acid it is difficult to nitrate under normal condition. So for this reason you will have to add alkali to these nitro derivative salts, and then you will get yellow to orange colored derivative. So similarly, in this table you will find uh, 10, 10 tests for the detection of different amino acids. For example, Milan test used to detect the amino acid that is called tyrosine. In the same way, Pauli's test used to detect the imidazole ring in the sample. So the amino acid which generally has imidazole as its side chain is known as histidine, right? And in the same way, through sulfur test you can detect the important sulfur containing amino acid, that is cysteine. And in another way, through Sakaguchi reaction you can detect the amino acid which has guanidino group in their side chain, and that is arginine. So this is very important table, so just go through it, and after that you will learn more about the detection of different amino acid by these reactions. Now I would like to talk about the polymers of amino acid, the acids that are peptides and proteins. So biologically occurring polypeptides ranges from very small consisting of 2 or 3 to very large consisting of thousands of linked amino acid residues. So in this diagram you can notice when few amino acids bond together by a covalent bond, that is known as peptide bond, uh, it it forms peptides, also known as oligopeptide. But when very large number of these amino acids join together by peptide bond, it forms polypeptide or protein. So peptides are distinguished from protein by their shorter length, right? So peptides is a short chain of amino acids. The amino acids in a peptide are connected to one another in a sequence by bonds called peptide bond. Now let's talk about peptide bonds. So just observe this peptide, that is dipeptide, and here two amino acids are covalently linked with each other through amide linkage, that is CONH linkage between two amino acids, and this amide linkage is known as peptide bond. Such linkage is formed by the removal of water from alpha amino group and from one amino acid, and alpha amino group from one amino acid, and alpha carboxyl group from another amino acid. So peptide bond formation is an example of condensation reaction. As you see here, these amino acid still leaves free amino group at one end and free carboxyl group at another end of dipeptide, and hence the reaction would be continued by adding more amino acid at both the ends, and this will lead to increasing the length of the sequence of amino acids. So this is the structure of pentapeptide, in which 5 amino acids, that are serine, glycine, tyrosine, alanine and leucine, are linked with 4 peptide bonds, that is 1, 2, 3, 4, right? And each amino acid unit in a peptide is often called as amino acid residue. So here amino acid residue means parts of amino acid left over after losing the element of water during the formation of peptide bond. So in a peptide, the amino acid residues at the end with free amino group is the amino terminal residue, or you can say it as N-terminal, and the residue at the other end, which has free carboxyl group, is known as carboxyl terminal residue or C-terminal. Here you will have to take notice of one thing, that when you have given a sequence of peptide like this, then amino terminal or N-terminal will be placed at left, and carboxyl terminal or C-terminal will be placed at right. So by this way, the sequence of amino acid will always start with amino terminal end. Now let's discuss about, so here I want to explain one more thing, that when few amino acids join together by peptide bond, it forms oligopeptide, and when many amino acids join together by peptide bond, it forms polypeptide or proteins. Now let's discuss about the acid-base property of a peptide. For this, let's examine the structure of this oligopeptide. As you see here, glutamic acid and lysine are added to this chain, and we know that the molecule of water must be eliminated during the formation of peptide bond, and the portion of each amino acid now known as amino acid residue in this oligopeptide. Here alpha amino group and alpha carboxyl group of non-terminus amino acids are covalently joined in the peptide bonds, right, and which don't take part in ionization, and thus, so as I told you earlier, since these are present in the middle of this chain, so that's why they cannot contribute in the total acid-base behavior of the peptides. However, the R group of some amino acids like glutamic acid and lysine can ionize, and in a peptide these can contribute to the acid-base properties of polypeptide or oligopeptide. So here we can say that in a polypeptide or in an oligopeptide, the side chain of the side chain of different amino acids generally take part in its acidity, and it takes part in its acidic property and basic property. Now I would like to discuss about the classification of protein. Proteins can be broadly classified into two ways. First way of classification of protein is based on their structure, and the second way of the classification of protein based on their function. So on the basis of structure, protein can be classified into four classes. First is primary structure of protein, second is secondary structure, third is tertiary structure of protein, and fourth type of protein structure, quaternary structure of protein. So let's first discuss about the primary structure of proteins. It is also considered as the simplest structure of proteins. Primary structure of protein is long linear chain of amino acids joined together by peptide bond. So this is the only long linear chain of amino acid. So this is also the first protein structure which only consists peptide bond. Since this is the phase of protein formation, and so they are normally, so this is the first phase of, so since this is the first phase of protein formation, so they are normally non-functional. But here is an exception, that is insulin. Insulin is the primary protein, but it is specifically functional. Otherwise all primary proteins are non-functional. Now let's discuss about the secondary structure of proteins. So the secondary structure forms due to the formation of hydrogen bond. So suppose these are the two amino acids here, and they forms hydrogen bond, and by this way these two amino acids come close to each other in this polypeptide, and this generally is responsible for the formation of 3D structure. So the bond involved in the formation of secondary structure of proteins are, first is peptide bond and second is hydrogen bond. Now there are two types of secondary structure of protein. First is alpha helix and second is beta pleated sheet. In alpha helix, H bond, that is hydrogen bond, forms between amino acid of the same chain. Sorry. But in beta pleated sheet, hydrogen bond forms between the amino acids of different chains. Alpha helical structure is found in keratin and beta pleated sheet structure is found in silk fiber. So you can say that the secondary structure of protein predominantly found in structural protein. So beta pleated secondary structure can further be classified into two types. First type is parallel beta pleated sheet and second is non-parallel or anti-parallel beta pleated sheet. In parallel beta pleated sheet, the polypeptide or strands all run in one direction. But here you can notice, in parallel beta pleated sheet that the direction of these two polypeptides these two polypeptides run in the same direction, that is N to C. But in anti-parallel beta pleated sheet, these two polypeptides run in different direction, right? So this is the main difference between the parallel and anti-parallel beta plated sheet structure of secondary structure of protein. Now I would like to discuss about the tertiary structure of protein. So this structure of protein is final and stable three-dimensional shape of protein in order to function properly. For tertiary structure of protein, important bond involves are peptide bond, hydrogen bond, uh, ionic bond, hydrophobic interaction, and disulfide bond. The tertiary structure of protein is the arrangement of secondary structure into this final three-dimensional shape. So tertiary structure of protein are generally functional protein, and almost all the enzymes generally has almost all the enzymes have generally tertiary structure of proteins. So now I would like to discuss about the quaternary structure of protein. So many proteins are made up of single polypeptide chain, and they only have three levels of structure, that is primary, secondary and tertiary structures, and I have discussed about all of these. However, some proteins are made up of multiple polypeptide chains, and when these polypeptide chains come together, they give the protein its quaternary structure. So examples are hemoglobin and DNA polymerase. So hemoglobin carries oxygen in the blood and is made up of four subunits, and that is two each of alpha and beta types. And DNA polymerase is an enzyme that synthesizes new strands of DNA, and it is generally composed of 10 subunits or polypeptides. So same types of interaction that contribute to tertiary structures also hold the subunit or polypeptide together to give quaternary structure. And what are those interaction? Those interaction are peptide bond, hydrogen bond, ionic bond, disulfide bond, and hydrophobic interactions. So this is all about the quaternary structure of the proteins. Now I would like to summarize the different structure of the protein. So initially, primary structure of the protein forms, which is the simplest structure of amino acid. After that, when within this primary structure a protein, hydrogen bond forms between amino acid within or between the two polypeptide chains, then they come close to each other and form secondary structure. And secondary structure are of two types. First is alpha helix and second is beta pleated sheet. And after that, in the next step, these secondary structure of proteins again folds with the help of different bonds, strong and weak bonds like covalent bond, hydrogen bond, disulfide bond, ionic bond, and hydrophobic interaction. And after folding, it forms three-dimensional structure, and these three-dimensional structure or tertiary structure of proteins are functional proteins. Now in the next step, or you can say in the last, when more than two polypeptides come close to each other, they form quaternary structure. So this is all about the different structure of the proteins. So if you have any query, please let me know about your query by phone, message, or so. Thanks to all of you, and bye bye.